Hi, everyone. Thank you for joining us for Remember the Girls' third webinar of our 2021 webinar series. Remember the Girls is a nonprofit organization dedicated to supporting and advocating female carriers and patients of X-linked disorders, a group overlooked by the medical profession. I'm Taylor Kane, the founder and executive director of Remember the Girls and the moderator for today's event. The topic of this webinar is titled, Why Are Some Carriers Symptomatic While Others Are Not? A specialist explains X-linked inactivation and other factors related to the physical symptoms suffered by X-linked females. We'd like to thank the sponsors of our 2021 webinar series, Horizon Therapeutics, Bluebird Bio, Ultragenyx Pharmaceutical, and Sanofi Genzyme. As for some background, X-linked genetic disorders are caused by an abnormality on the X chromosome. Since males, XY, have only one X chromosome, they develop the disorder. Females with two X chromosomes, one which contains an extra unaffected copy of the gene, have traditionally been to be just carriers who are completely unaffected by the disorder. For most X-linked disorders, this is simply untrue. In addition to the fact that female carriers may pass the disorder along to their offspring, many do develop physical symptoms of the disorder, sometimes severe. Often, however, they fail to connect these symptoms with their carrier status, subjecting themselves to years of physical problems that they should not have to tolerate. There's long been a gender bias in medicine. For decades, patients enrolled in clinical trials were predominantly male. Most of the studies that did include women did not report their results by gender. And although this has begun to change in recent years and medicine is recognizing that diseases and treatments affect the sexes differently, there is still an enormous lack of research and understanding when it comes to female carriers of X-linked disorders. For most X-linked disorders, the medical profession is just beginning to recognize that females are not just carriers. And while it's extremely important to continue searching for treatments and cures for males with X-linked disorders, it's also important to study the effect these disorders have on female carriers too, so that they have the opportunity to live a full and healthy life. So today we are joined by Remember the Girls member, Laurie Hayes, who will be sharing her journey as a symptomatic female with ALD. And after her presentation, I will introduce Dr. Barbara Migion. It's French. Tried my best there. An expert who is going to be sharing who will be explaining why some females with X-linked diseases experience symptoms. And I will give a more full introduction once we move on to her presentation. But Laurie, take it away.
Taylor asked me to speak. I have been very fortunate to have met her, and she is doing amazing work for women. So I am very, very thankful to know her. I am 52 and I am a symptomatic woman. I have ALD. I have a whole family history, but in particular, my mother has it and two of my brothers do, and my sister does. So out of 5 of us, only one is unaffected. We had always been very concerned about my brothers. And what the future would look like for them when we all went to Baltimore to meet with Dr. Moser 30 years ago. We were all tested and he told me that I was a carrier. I had nothing to worry about. Perhaps when I would be in my 60s, maybe I would have to use a cane. And all along, we were concerned about my brothers and experimental treatments and studies. And well, fast forward now, and about 5 years ago, I started having symptoms, and then they got progressively worse at a rate that was pretty fast. So I went from being on the beach 5 years ago and walking around to mostly being in a wheelchair. So it's been quite a journey, and it definitely has had its ups and downs. For females, well, for anyone with ALD, there are definitely some commonalities with symptoms, and bowel and bladder problems are at the top of everybody's list. So that is very challenging, and that affects me and my siblings. Walking is a huge struggle. But I would say that I am very fortunate to live right outside of Boston. And so I have access to amazing doctors who have worked and continue to work to focus on women and the struggles that they are going through. And I think my biggest message is that you have to be your own best advocate, and you have to ask the questions. And thankfully, my doctors understand that women do have the disease. They're not just carriers. And I'm fortunate to have that. However, I've spoken with so many women across the country that don't have doctors that understand what they are going through. They don't believe it. They think it's in their head and that they're making up some things. And it's really scary for those women. So I think if I only make one point, it's that you are your own best advocate and you can't ever stop. And don't be afraid to try new things. And I think it's really important to surround yourself with a ton of support and positive people, because it's very easy to get down when your life changes so dramatically, so quickly. And you should never feel badly about having a bad day if you are experiencing symptoms. And then I think the scary thing is for women, when they hear from people like me, they worry that this will happen to them. And unfortunately, there are just no guarantees who experiences symptoms and who doesn't. And, I am doing the best that I can, and I am very fortunate to have a loving husband and two amazing girls who always, always look out for me. So it's not the end of the world. It's just something different. And it just makes my life more of an adventure than it was before. So I am thrilled to be part of the Remember the Girls community and the ALD community. And it has been life-changing for me to find support in these two different communities. So I think for everybody who's listening right now, you really have an amazing network here with Taylor and all of the women who belong. And just never stop asking questions and never stop looking for an answer and trying whatever you can. So that's go ahead. Sorry.
Thank you so much, Laurie. That was amazing. And I'm one of the people who is so appreciative of all that you've done for ALD females, and speaking at our ALD events and just being such a powerful advocate and showing that you are a firsthand example of the fact that women clearly experience symptoms, and they're not just the mild symptoms that we might have been told to think, and that they can be life altering and they can be severe. But I appreciate you sharing, and I'm proud to advocate aside you as well. Thank you.
Thank
And
you.
also, if anyone has any questions for Laurie, you can send those in the chat. We'll address the questions at the end after our second presentation, but just send them and we will get to those. Okay, great. So now we will move on to Dr. Migion's presentation. Okay, and now I'm just going to read some information. Barbara R. Migion is a member of the McKusick-Nathans Institute of Genetic Medicine and a professor emerita in the Department of Pediatrics at Institute of Genetic Medicine at Johns Hopkins School of Medicine. After receiving her bachelor's degree from Smith College and medical degree from the University of Buffalo, Dr. Migion obtained clinical training in pediatrics at the Johns Hopkins Hospital. She has postdoctoral training in endocrinology at Harvard and in genetics at Hopkins. She is board certified in pediatrics, biochemical genetics, and cytogenetics. Dr. Migion was the founding director of the PhD program in human genetics and molecular biology at Johns Hopkins University and continues to teach in that program. She's been a member of the genetics, mammalian genetics, and genome study sections at the NIH, the boards of the American Society of Human Genetics, the DNA Methylation Society, the Epigenetics Society, and the editorial boards of Trends in Genetics, Cytogenetics and Cell Genetics, Somatic Cell Genetics, Cancer Research, and Epigenetics. Since the beginning of her research career, all of it at Johns Hopkins, Dr. Migion has been working to understand the molecular mechanisms involved in the X chromosome inactivation in human cells and the clinical consequences of a single working X chromosome for both sexes. She has published more than 150 papers on the subject. Her book, Females Are Mosaics, X Inactivation and Sex Differences in Disease, has been published by Oxford University Press. She's written a second book, American Science by View from the Bench. Her postdoctoral fellows and graduate students have become leaders in their various fields of human biology and genetics. Dr. Migion was the sixth woman to achieve the status of professor in the medical school and has worked to promote the role of women in this institution and elsewhere. She has received the Marion Spencer Fay Award for her significant contribution as a woman physician and a Lifetime Achievement Award from the March of Dimes at the national meeting of the American College of Clinical Genetics and Genomics. So as you can tell, Dr. Migion has had an incredibly accomplished career. And even though she would say that she's retired, she is still continuing to research. And of course, she's here speaking with us today to provide us some of her expertise about X inactivation and, the mechanisms that cause females to have symptoms of X-linked diseases. So we're so grateful to have her here today. And she will be sharing her presentation. So feel free to share your screen, Dr. Migion. And if anyone has any questions, feel free to send those in the chat or in the Q&A section, and we will get to those. Thank you.
Thank you so very much, Taylor, for that introduction, and Lori for your lovely presentation. I'm interested in the mechanism that has to do with compensating for the fact that males have one X chromosome and females have two. I've worked on it all my life. I had the opportunity to work with Hugo Moser on ALD near the beginning of my career. And we made some interesting observations at that time that I want to tell you about. In addition, I had the opportunity to go to one of the camps. And so I met some of the heterozygotes for ALD who were in wheelchairs. It sort of shocked me at the time. But I thought I knew the reason why, and I'll tell you a little bit about that. So I'm going to talk to you about why some heterozygous females, and they're carriers, but you're heterozygotes, and I want you to know what that means, why some of you manifest your X-linked diseases while others do not. And let's see. Okay. I'm going to start by explaining some simple genetic principles so that we can be all on the same page. In case you have some problems with understanding what I'm talking about and would like to read more, I have written about it. And as Taylor told you, it's Females Are Mosaics, and it's about X inactivation and sex differences in disease. I am a basic scientist, but I'm interested in sex differences in the way diseases present. And so I'm not a practicing physician anymore. I used to be, but not anymore. So yeah. Can you see my screen without all of this? Women have the possibility of having two different forms of any of their genes. And those different forms of a gene are called alleles. Homozygote women have 2 copies of the same allele. Most often they're normal, but some homozygotes can have 2 copies of a mutant allele, and they would obviously manifest the mutation if that were the case. But heterozygotes have 2 different alleles. Now, one of them can be abnormal, or both of them can be normal in different forms of normal. But they're different from one another, and that's why they're called heterozygotes. Now, one differs from another or doesn't work well because of a deleterious mutation. Now, those heterozygotes with one deleterious allele are called carriers. So all of you are heterozygous for mutations on the X chromosome. And I want to just go over with you what a gene is, what an allele is, what is a mutation, and what is a deleterious variant. These days, people don't want us to use the word mutation. They prefer that we use deleterious variants to explain the, the results of a mutation that ends up producing a disorder. Now, you know that the cell has a nucleus and a cytoplasm, and within the nucleus of the cell are the chromosomes. And these are the chromosomes. They're really duplicate copies of a single strand because we see them as they've already replicated, ready for cell division. And one of each goes to the daughter cells. And so you end up with having both new cells have the same chromosome. Along the chromosome are the genes, and they are in order along the chromosome, and they can be different sizes. Some genes are quite large and others are small, but they're all made up of a code. It has 4 bases: guanine, cytosine, thymidine, and adenine, and the order of them specifies the amino acids that make up proteins. So the building blocks are all the same. Now, I'm showing you human chromosomes, and they've been stained for specific DNA content. So each one is stained a different color. You can do that, and it's actually glorious to look at under the microscope. And as you will notice, they come in pairs. And the reason they come in pairs is so that there's a pair of genes for almost every trait. And that's a good thing because genes are made up of alleles. And you can see it best if you think of eye color genes, because we can have a brown gene or a blue gene or an emerald gene or a gray gene. They're all normal, but they're different from each other. And so someone who has these 2 is considered a heterozygote. Whereas if you have 2 genes for brown, you're homozygous. Some alleles work and others do not. Here you have different hair color, and you can have a mixture of several kinds of genes that will give you different hair color from someone else who has a different pair of alleles. Now, we said that these human chromosomes generally come in pairs, and pairs protect you against the effect of mutations. Because if you have a mutation in this chromosome, then the normal copy of that gene is going to be okay on that chromosome. And so you get a sort of a blend of the 2 in each cell. And 50% of normal activity is usually enough. So that if you have a mutation in one of those genes, it doesn't necessarily show up, okay? And you have to have sometimes very low amounts of the product of the gene in order for it to manifest in any kind of way. Now, cells sometimes make mistakes when they're copying, gosh, their DNA, and these mistakes are called mutations. And mutations are actual changes in the genetic material. Some mutations may be deleterious, but often they are not. They disturb the code for the gene, but that doesn't seem to make much difference in terms of the end product. But those that interfere with the function of the gene are deleterious, and that is what a mutation is. Now, the exception to chromosomes coming in pairs are the X and Y chromosome that I think you can see here. Can you see it?
Yes.
Well, anyway, that is a problem immediately for a male, you can imagine, because any mutation that happens on the chromosome that is responsible for loss of a protein product can be deleterious for him, can be devastating. And I just want you to know that the male has a Y chromosome in addition to his single X, which always comes from the mother. The Y comes from the father. And there's a big difference in the gene content of those chromosomes. These have at least 1,100 genes on the X chromosome, and there are about 100 genes or so on the Y chromosome. And it's even worse than that, because these genes on the Y chromosome are differentiated genes. Many of them, the majority of them, have to do with fertility in males. They determine that you have a testes, and they determine that the sperm are able to fertilize eggs. And so there are lots of fertility factors, and they are really sex-related genes and male sex-related genes. On this chromosome, however, there are very few genes that specify anything to do with sex. They instead have everything to do with bones and brain and skin and liver and kidney and all that kind of function. And they're pretty much like all the other chromosomes in terms of the kinds of traits they carry. So there are very few fertility factors. They don't specify sex characteristics, and they're like autosomal genes. And it's lovely that females have two of them, whereas males have only one copy of them. And I'm going to tell you that your two X chromosomes really do make you not only as good as males, but superior to them in terms of health issues. Even though some of you are feeling the effects of X-linked mutations, you have to think about all the effects you don't have and that you won't have, because what is true about the X coming in pairs in females, but not in males, is that it does contribute to our health. And we do live longer, and you know that. We'll show you that in a little while. The fact that there's one X in males and two X's in females necessitates some form of dosage compensation. That's a big word for the X chromosome. But I have to tell you that any organism that has an XX, XY kind of sex determining mechanism has the same problem, whether you're a worm or a fly or a fish or whatever you happen to be. if you have an XXXY kind of sex determining mechanism, you have to do some compensation. Now, it's done differently in different species. For instance, in Drosophila and flies, what they do is to jazz up this X in males so that it equals the two X's in females. That's a clever way to do it. And it works quite well because you then have everything in pairs, essentially. In humans, we don't do that. Instead, what we do is to inactivate one X chromosome in each cell. So we have both X chromosomes, but the fact is we can't transcribe both X chromosomes. And we know that because we never saw a female who had two X chromosomes. Every female has to somehow or other inactivate one X chromosome or she doesn't survive. And so I did want to call your attention here to the pseudoautosomal region on X and Y chromosomes. That's the part of the chromosome that's identical on X and Y. And there are about 20 genes or so. It's not very large. It's at the tip. But the reason it exists is that it helps in chromosome pairing and segregation during meiosis, which is when you make the sperm and the eggs. And so it helps to have some part that's identical. Here you're pretty identical all the way through, but in the male, you have no identity, very little identity between the X and Y. And so that these particular genes, which are expressed on every chromosome, whether it's inactive or not, and they're important genes. There's some stature genes there, which helps explain what happens when you end up with only one, or you have two X's in a male. They're taller. If you have one X in a female, you're shorter. So that particular gene is quite important and in that region. So I wanted to tell you that we know that you have to inactivate one X chromosome because if you have tiny, tiny extra ring that's expressed, it's a little ring chromosome. And we see those individuals quite often. Look how tiny it is compared to the size of an X, which is like this in general. And when you have that, you end up with girls that are quite similar in there, even though they're not related in their facial characteristics, and they're quite abnormal and quite severely retarded. So even if you express a tiny bit of the second X, you're in big trouble. And so you can understand why you need to turn off one X chromosome in each cell. Now, I wanted to introduce you to Mary Lyon. She's the one who proposed X inactivation. She was a mouse geneticist, and she was looking at her mouse mutants, and her hypothesis is based on the studies of a mouse that's called mottled. I always tell my students, because they have to understand that her hypothesis didn't come out of nowhere. It was based on prior studies of others. She had to know that you could survive with one X chromosome as Turner females do. She had to know several observations that I won't go into here, but she did come up with this hypothesis, and she extended it to humans the next year. Now, this is a mottled mouse. It's all white instead of having the skin color of a normal mouse, which is brown. And she didn't think that was too unusual. She knew the gene was on the X chromosome. But she looked at the heterozygote And she thought, well, if you have two genes on two X chromosomes, and you have one model gene, you have a normal gene, you should have a blend of the two colors. So it should be sort of a tan animal. But instead, she saw that there were patchworks of black, a normal mouse color, and the white. And she said, well, what could be going on there? And she came up with the idea that the model gene was on this white chromosome here. The normal gene is on the brown chromosome, normal skin color. And she thought that in each cell, only one X chromosome was working, and one was made to be heterochromatic or somehow or other didn't function anymore. And in cells in which you saw the brown color, it was the white modeled mutation chromosome that was inactive, whereas in the cells in which you see the white color, then it was the normal gene that was inactive. And so that's how she came to her hypothesis. Does that make sense to you? Yes. Okay. So it was, I came to Hawkins as a pediatric resident, but I ended up coming back after my endocrine fellowship and getting married, and to study genetics with a man named Barton Childs, who was one of the first human geneticists. And he started, he and Victor McCusick started human genetics at Hopkins, he in the Department of Pediatrics and Victor McCusick in the Department of Medicine. And Mary Lyon didn't have any experiments to show that what she was saying was true, but they figured out a way to show it. And they, yes. I don't know how to get rid of this thing. Can you see that?
The heterozygous skin cells slide.
Well, no, you were screen sharing, stop share. No, you don't see that. And mute. No. Okay.
No, no.
All right. So, good. What they did was to take cells from women who were heterozygous for the enzyme G6PD, glucose 6-phosphate dehydrogenase. Now, most Caucasian people have just G6PDB, but people of African descent often have G6PD-A as well as G6PD-B. Both of them can be normal. It's just like having blue and brown eyes. But you can figure out a way of identifying the B form of the enzyme versus the A form of the enzyme. And they used a gel in which one migrated farther than the other so they could distinguish the two. And when they took the skin cells and grew them in culture just together, they got both A and B, which is what you would expect, because even if some cells were expressing A and other cells were expressing B, or if both cells, you know, all cells express both A and B, they didn't know. But then they cloned the cells, which means clone here means taking individual cells from these carriers and putting them in a dish, maybe 10 cells per dish, and 3 will grow up, and you let them grow. And what you have here are all the descendants of a single cell. So they all came from the same original cell. And in this case, they all came from the same original cell. And then when they grew these clones up so that they could analyze them, they found that some of the clones expressed A and some of the clones expressed B. And so that was proof, very important proof, not only that some cells expressed A and some cells expressed B, but that it was a fixed process. So as the cell grew, it couldn't change its type. It had to stay the same. Does that make sense to you? Do you see
Yeah.
that?
Yeah.
So it said that clones express A or B, and they don't change as they grow. They keep whatever they were meant to be stays put. Okay. So I wanted to show you just because many of you, I think, are interested in ALD. We did that same experiment using cells from individuals who had both G6PD and ALD variants. We could find them. And so we could tell what the clone was on the basis of either G6PD or ALD. And I looked at the G6PD. I would send them in an unknown fashion to Hugo Moser, and he would analyze them for their ALD type. And then we came to the surprising, I mean, it really surprised us that uh, A, in the case that we were seeing, was always associated with the ALD variant, and B was always associated with the normal ALD allele. And that's because they're both together on the chromosome. They're both at XQ28. They're genes that are linked to one another so that they don't change. We had thought that they might have changed, but they didn't. So we knew that they express G6PD A or B, but in addition, they expressed either the mutant ALD or the normal ALD. And then we could go on, and I'll tell you that we saw with the ALD as opposed to G6PD, there were too many ALD clones. We expected to see more normals, about 50%, because if you were choosing one chromosome or the other, it should come out about 50%. Well, what we did was to take the age of the heterozygote and the number of clones analyzed and the percent mutant ALD clones. And what you can see is that although early and I would say 70 and 30 are not much different than 50%. But when you start getting someone at age 23 with 100% mutant clones, this is the number of clones analyzed. Here we repeated one, so you can see the answer comes out pretty much the same. Here in a person who was 59, 100% of the clones showed the mutant form of the disease. And I have to tell you that this is completely unusual. And it's the only disorder that I know of where the mutant gene seems to have some kind of advantage over the normal gene, and that as the carrier grows older, she gets more and more mutant cells, which is an explanation, Laurie, of why you start to manifest the disorder. And it may be independent of your brothers because you're doing something that they're not. They don't have this selective advantage of the mutant allele, fortunately, but you do, and that's why you are starting to do that. So here is a simple scheme of X inactivation. We see the sperm and the oocyte coming together, and here's the X from the oocyte in red and the X from the sperm in blue. And they both function very early in the zygote. And then about the time of implantation, when the egg arrives in the womb, that's when random X inactivation occurs. And it is random when it occurs. It doesn't pay any attention to what it is. It just occurs one X or the other. And so that you end up with the inactive X as a bar body it's a heterochromatic chromosome that doesn't get transcribed. And then you have this process of mitosis, and that's the cell replicates in every tissue. We need to grow, and we do that during embryonic life mainly. We do that until puberty, and then after that, we stop growing. But we are growing, and the thing that is quite interesting is that whatever it was at the time when the X becomes inactivated stays with it through all that cell division, so that whatever happens here is fixed and doesn't change. It's a very stable kind of inactivation. But the result is that you end up with two kinds of cells, one in which the mother's X is working and one in which the father's X is working. That's clear enough?
Okay. Yes.
So I thought you might want to know a little bit about how an X gets inactivated. And there is this gene right on the X chromosome, about in the middle of it. And it doesn't make a protein like most genes do, but it makes a transcript. And that transcript is able to spread down the chromosome from both sides. And what it does is to attract all the epigenetic factors that inactivate a chromosome. And so it ends up with as being a condensed, non-transcribable chromosome. So the genes are inactivated. And that is called exist for X inactive specific transcript. It's only transcribed from the inactive X, and it's a very potent gene. If you take this gene and put it into a Down's syndrome cell, mouse cell, you can turn off the extra 21 because it's a potent inactivator. Once you put it into a chromosome, it has to inactivate the genes. Okay. And I thought you might like to see a picture of it. We can stain chromatin for being active or inactive. You can see that the centromeres often of the chromosomes are red. But here's the active X and here's the inactive X. And you can just see the difference between the two chromosomes because this doesn't have the materials, the epigenetic materials that allow it to be transcribed. You remember that we have the same genes in every cell, in every tissue, but only some of them work. And that's because of epigenetic factors that come in and inactivate certain genes and allow others to be transcribed. But this chromosome is pretty much inactive in every cell. I thought you might like to see what the significance of that is. I don't know why. Sorry. I'm showing you the colon tissue in your colon, and it's stained for G6PD. And you can see that this is from a heterozygote, and you can see that the unstained ones are the mutant cells and the stained ones are the normal cells. And you can see that it has an interesting pattern because cells stay together during their migration. And so that often is the case. And so you can see these patches of normal cells and those patches of mutant cells. So you are that mosaic in every tissue. And that's a good thing, because these genes can share their goodies with some of these. In this case, they're not, but it doesn't make much difference. But in certain cases, you can share the product of the gene that makes it with the gene that does not. And that's the good thing that we have going for us. So the genetic consequences of X inactivation is that we have a single active X in both sexes, females as well as males. But the good thing is that females are mosaic. They have some cells with their mother's X active and other cells with their father's X active. So what's the male disadvantage? Well, it's obvious, isn't it? Males have only one copy of their X-linked genes, and that's not a good thing. And I'm just showing you the sex ratio of mortality at various ages in the United States. And they're usually behind in years. They don't get this data out very rapidly, but I just wanted to show you both 2008 and 2014 so that you could see that they're pretty much essentially the same. It doesn't change. This is in the United States. It doesn't change in most countries, except if you do some kind of selection of embryos to get males, as they do in some countries. But I think you can see that at every age, from 0 to 1, 1 to 4, 5 to 14, this is the age when males do stupid things, ride motorcycles, and so that more males die than they would normally do it. But you can see that until you get to be about 80 years of age, the amount of males to females that are dying are unequal, and always more males are dying at every stage. So we may feel sorry for ourselves as females in some ways, but believe me, males have it much worse in general, because they are subject to a lot of disorders that we are not. And I just wanted to show you this. 95% of females, because there are always a few females that manifest everything. And we'll talk about why they do in a minute. But this is Duchenne's muscular dystrophy, and you have gradual muscle destruction, and males die in their teens, and females are usually not affected. This is a severe immunodeficiency of B cells. And again, the males are quite deficient, and some of them can't live a normal life. These days, they've been doing some transplantation in order to try to help these males, but females are not affected. And hemophilia is another one. There's factor 8 or factor 9. Both of them are made on the X chromosome. Severe bleeding in males. Females are usually not affected. Now, when we get into something like ornithine transcarbamylase deficiency, where males die in utero or close to the time of birth, or they did do that until they can be treated a bit now. But it was the females that had the disease, and they had meat intolerance and had too much ammonia in their tissues. And they were the ones to show the disorder because the males died. And the same is true of incontinentia pigmenti. males die in utero, and therefore it's the females that manifest the disorder. Okay. So you can understand why these females are manifesting, because the males have all died. So males bear the brunt of the problem. Now, female advantages, of course, are mosaicism. And I'm going to show you a little bit more about this because the size of the clones in us varies. This is in the placenta, and the patch size is very big there. A whole clone of villus in the placenta, and the villus is very necessary to transmit nutrients across the placenta to the fetus. And some of them are completely mutant. This is a Fragile X heterozygote and stained for the Fragile X protein. And this is mutant because it doesn't stain. And this is wild type, normal, because it takes the stain. And you can see that the patch sizes here are very large. But look at the brain. And you can see that they're very, very small. And so it depends on what tissue we're talking about. And these closeness in the brain has a lot to do with how we perceive color. We have much better perception of color. It's not a coincidence that most interior decorators are female, because they see colors that males don't ever see. So we have a lot of things. We have a greater variety of proteins that we make that males don't have. So it's a good thing that we're mosaic. Now, I did want to say that there's a sharing of this is the normal gene, the cell that expresses the normal gene, and the white is the mutant, and you can see that they're very close, and so that you can expect that there will be transmission, and there is transmission of gene products from cell to cell, as long as they can be transmitted. They're transmitted through blood. If the product can be transmitted through blood, it will be. If it can be transmitted through the intracellular junctions that cells make with one another that transport small molecules from one cell to the other. And if they can't share, then what happens is often the cell that is abnormal is overgrown by the cell that's normal. So this unequal ratio of cells with either maternal X or paternal X taking over is called skewing of X inactivation. So we start this way with any clone. And you can see that the clones with the paternal X and the clones with the maternal X are inactivated at about a 50% ratio. I'll tell you more about that ratio in a minute. But once that happens, then there's all sorts of possibility. There can be cooperation between the two cells. I'm sorry. There's some disorders where, unfortunately, the cell gets a little schizophrenic with two signals coming out, and it doesn't work very well. And often females, for rare diseases, and some of it is I would say that there are minor diseases. There are about 3 or 4 that are known where there is a problem, and that females will have more of a problem than males because the males can substitute another product for the one that they don't have. But the female who has one normal and one abnormal ends up with metabolic interference. But often what happens is cell selection and elimination. And one of the real reasons that there is cell selection, there are many of them, and I've shown you an X chromosome with some of the genes along the chromosome that we're going to use as models. And you can have, when an activation occurs initially, it can be skewed. And although the majority of people have 50%, 50% is a bell-shaped curve around a mean of 50%. And so some people can be 30% versus 70%. It's like flipping a coin, and that's the way it turns out. And a few unlucky people will be down where they flip out at 5% of one and 95% of the other. And if it, of course, is 95% of normal, that's always good. But if it's the abnormal one that's 95%, then obviously female is going to express that mutation. So one of the real first reasons is if some of the exist gene doesn't work well. That's primary random, non-random inactivation. In that case, what happens is that gene can't ever be on the inactive X. It's always on the active X. And so they end up being quite like a male. They have only one population of cells, all with the same chromosome active. But that's not the worst thing. You're just a male at that point. There's also, we talked about chance skewing, and here's the curve that I'm talking about, because it is 50%, but you can see that's the average, and that 30% and 70% are within one standard deviation of the mean. And so they occur frequently, and you can see the numbers of people that would be affected there. But you get down to this ends of the curve, and that you're within 2 standard deviations. Here's 2, 3, 4, and you end up with people who either have too many of one cell type or too many of the other. So secondary skewing is probably the most that we see. And I'm just giving you some disorders where we see secondary skewing. And I must say that ALD is the only one where the mutant cell has the advantage. All the others, it would be the normal. So I'm sorry about that. And I have no idea why having extra long chain fatty acids in your cell membranes does the cell some good, but it seems to like it better than it does the normal variety. So what I want to say is, that if you have X autosome translocations or you have deletions of your chromosome, that chromosome is often inactivated. So whatever genes are on that chromosome are going to be inactive. So it is a growth advantage of some alleles and some chromosomes. But I think you will get the fact that if you have skewing favoring the normal allele, then it can ameliorate the disease in carriers, right, in heterozygotes. But if you skew in favor of the mutant allele, then you create what's called manifesting heterozygotes. And that's 5% of normal females, regardless of what genes are on the chromosome. But if you have ALD, then you have, unfortunately, a greater chance of being a manifesting heterozygote. I haven't talked about it, but of course, a lot depends on the nature of the mutation you have, the deleterious variant. Some of them make you lose all activity of the normal gene, and others retain some activity. And we are finding that as little as 10% normal activity is often enough to present the manifestations of the disease. And that's very hopeful because in cystic fibrosis, for instance, if you can raise the level of the normal gene product to 10% in a cell, that's enough to correct the problem. So it means that a lot of carriers of that disorder are going to be able to have normal lives. And I must say this is a very hopeful time in the history of genetics because we are seeing more and more possibilities of correcting defects. And I think we can look forward to it. Within the next 20 years, we're going to see enormous amounts of corrections of mutations that we thought were difficult to deal with. And so you just have to keep in there, keep working at it. But in general, it's good to be a female. It really is. So I think you can see that even identical twins are not identical, right? Because they do have differences in their skewing. And for some reason that we don't understand, identical twins are they're produced about the same time as X inactivation occurs in development, and that we see an awful lot of reports of one twin expressing a disorder that the other twin does not because of skewing in favor of the mutant allele in one case and not in the other. So identical twins don't even express the same diseases. Yeah. Well, I think that's what I have to say, and I'd be happy to answer any questions that you have that I
That
can.
was incredible. That was so informative. And you really touched on, so many points that I had been wondering about over the years, like why certain diseases like ALD, there seems to be more symptomatic females when different X-linked diseases, there aren't as many. So I also appreciate you breaking it down for us and explaining like all the scientific concepts. So it was really easy to follow. And I'm no speaking from my perspective, and I'm sure everyone on this call would agree, but I'm just so appreciative of all the work that you've done. Like it really makes me feel seen as a heterozygote with ALD, just knowing that there has been research on this and that, if this research continues, there will be a greater understanding of why we are often symptomatic. So just want to thank you for that, Dr. Mijion.
Yes,
So we do.
To say one thing about ALD is that I did listen to Jerry Raymond the other day. I think some of you know who he is. He's sort of taken Hugo's place at Hopkins, and he was talking about ALD, and it is a fascinating disorder because members of the same family don't necessarily express the disorder in the same way. And there must be some modifying genes that are involved, but they haven't been found yet. So there's a lot of work still being done, and you will benefit from it one of these
That's
days.
great. Yeah, looking forward to that for sure. So we do have a few great questions. The first one will be for Laurie, and we are a little short on time, but we'll be able to get to a few of them. I also, if there are more questions for Dr. Migion, either I could email them to you, or I could.
Yes, I'd be happy to answer them.
Okay, great. So the first question is for Laurie. When did you first start to notice that you had symptoms of ALD? And were they there, but you like brushed them off, I guess, because of the assumption that you were just a carrier?
Well, I would say that for as long as I can remember, I have had balance issues. And my family just made fun of me and thought that I was clumsy. And that could be the case. I don't want to be that person who attributes everything to ALD. So I have no idea. Now I really have bad balance issues. I would say that I specifically remember having an incontinence issue when I was 38 because I remember it because I was in Puerto Rico with my husband and I had a problem. So I can say definitively that's when I started to notice it. Um, about 5.5 years ago, I broke my toe. And that doesn't seem like a big deal at all, but I was in a huge boot for a few months. And I used to exercise every single day and I couldn't. And I really feel like my lack of inactivity kind of, maybe spurred it on a little faster, or I don't know, maybe I'm just assuming that. But I would say in the past 4 years, since I was 48, things have significantly declined. So
Thanks for sharing. We have a comment from Jamie, who I'm sure you know, but she was mentioning how she's 54, and as of a few years ago, her symptoms have onset very quickly, and that she experiences a lot of falls as well. And I know in her case, they've also come on very quickly. And she now is using a walker, but still has struggles with falling, even with the walker. So one of the questions that we have, this is a great question for Dr. Migion. So do female carriers get the same symptoms as men, just to a lesser degree, or can their symptoms be entirely different? Would a treatment that works on like an affected male, say an AMN treatment in the case of ALD, necessarily work on a symptomatic female?
Well, that's a hard question. Generally, females do better no matter what. If they have it depends on the nature of the mutation, but at every state, milder mutations are manifested only by males. It's more severe ones, females start to manifest them. Whether they have the same or I think that's probably an ALD problem and probably due to some modifier gene and not particularly this process, which usually makes a woman better no matter what the mutation is than what she would be, what a male would be with the same mutation.
Right. Okay. I believe some recent research for ALD carriers and ALD women were showing that some of them experience dystonia. And as far as I know, I don't believe that men with AMN experience dystonia. So I know we were all interested in that because we were kind of told like, oh, women are just like less severe versions of men with AMN, but it seems that there could be things that we actually experience differently. So hopefully that research will continue to, yeah,
yes.
Right. So another question we have is so it's talking about just why females have a higher incidence of autoimmune disorders and wondering if this is because of the diversity of self-antigens.
No, remember I told you that that there was a pseudoautosomal region on the chromosome. And there are some other genes that are expressed from the inactive X. I didn't want to talk about it, but there are a few genes, about 15% of the genes. They aren't expressed well. They may have 10 to 30% of the activity of the gene on the other chromosome. But one of those genes happens to be the toll receptor 7. There are lots of immune genes on the X chromosome. And it turns out that women have much better immunity than males do because we have a little extra going for us in genes that are expressed from the inactive X. But we also have more autoimmune disease. And that's the price that we pay for having a better immune system.
Great, thank you. So just for our last question, and I believe in one of your research papers, you had mentioned MPS II Hunter syndrome. So the question is, what diseases have been researched to see if there are manifestations in females? And the person with this question was thinking of lysosomal storage diseases with missing enzymes, such as MPS II.
Yes. Well, there's a big difference between Hunter's disease and Fabry's disease. In Fabry's disease, heterozygotes often manifest, not as severe as males. They don't have it as severe. They don't have the onset as early as males do, but they can. Whereas in Hunter's, the heterozygote is literally very, very, very rarely affected, depending on whether she skews by chance or something of that sort. But the difference is that there's a sharing of the enzyme from cell to cell. And in Hunter syndrome, the enzyme works very well. The lysosomal enzymes participate in sharing their products, and it's a high uptake enzyme. And so that was one of the treatments of Hunter's disease in males, is to give them the enzyme and so that they can get it. But females do that naturally. They feed their mutant cells with the normal product. But with Fabry's disease, it's a very low uptake enzyme, and it's very difficult for it to go from cell to cell. And that's why there's a difference between that one and the other, although they're both lysosomal diseases.
That's incredibly interesting. Yeah, and of course, also that, as you spoke about in the presentation, ALD is the only X-linked disease that's known right now where the mutant cells favor, the body favors the mutant allele. So we're definitely not very lucky on that side of things, but I know we'll continue working and advocating for more research. And
I'm sure that the therapies that are worked out in males can be used in females.
Yeah, hopefully, because we do have a few different drugs that are being studied for men with AMN right now. So I know if there are a lot of females, with symptoms who hopefully could benefit from those drugs. But thank you both so much for taking the time today. This was an incredible presentation. And it will be recorded. So I will send you the recording once we've edited it. But if there are any other questions for any of our viewers that you would like for either Laurie, you can find her in our Are you still on Facebook, Laurie? Or okay. Yeah. I thought you deleted it for some reason. I'm not sure. Laurie's in our Facebook group. And then if you have any other questions for Dr. Migion, you can email those to me and I can pass those along to her. But thank you both for your incredible presentations, and thank you everyone for attending.